DNA is the hereditary material as it contains the genetic information. It is the most important constituent of a chromosome. The most widely accepted model of DNA is the double helical structure of James Watson and Francis Crick. They proposed the three-dimensional model of DNA on the basis of X-ray and diffraction studies of DNA obtained by Rosalind Franklin and Maurice Wilkins in appreciation on their discoveries on the molecular structure of nucleic acids. Watson, Crick, and Wilkins were awarded the Nobel Prize for Medicine in 1962. Chemical composition of DNA molecule. DNA is a large molecule consisting of billions of nucleotides and is also called as polynucleotide. Each nucleotide consists of three components: a sugar molecules, deoxyribose sugar, nitrogenous base. There are two types of nitrogenous bases in DNA. They are purines, adenine and pyridine, cytosine and thymine have phosphate group. Nucleoside and nucleotide. Nucleoside, nitrogenous base plus sugar. Nucleoside plus phosphate. The nucleotides are formed according to the purines and pyrimidines in the Watson and Crick model. DNA molecule consists of two polynucleotide chains. These chains form a double helix structure with two strands which run anti-parallel to one another. Nitrogenous bases in the center are linked to sugar phosphate units which form the backbone of the DNA. Pairing between the nitrogenous bases is very specific and is always between purine and pyrimidine linked by hydrogen bonds. Adenine links thymine with two hydrogen bonds, here double bond T. Hydrogen bonds. Cytosine links with guanine with three hydrogen bonds, C triple bond G. This is called complementary base pairing. Hydrogen bonds between the nitrogenous bases make the DNA molecule stable. Each turn of the double helix is 34 Armstrong. There are 10 base pairs in a complete turn. The nucleotides in a helix are joined together by phosphodiester bonds. Chargaff rule of DNA pairing. Yergin Chargaff states that in DNA the proportion of adenine is always equal to that of thymine and the proportion of guanine is always equal to that of cytosine. DNA replication is one of the basic processes that occurs within a cell. DNA molecule produces exact copies of its own structure during replication process. The two strands of a DNA molecule have complementary base pairs. The nucleotides of each strand provide the information needed to produce its new strand. The two resulting daughter nuclear cells contain exactly the same genetic information as the parent cell. DNA replication involves the following steps. Origin of replication. The specific points on the DNA where the replication begins is the site of origin of replication. The two strands open and separate at this point, forming the replication fork. Unwinding of DNA molecule. The enzyme called helicase binds to the origin of replication site. Helicase separates the two strands of the DNA. The enzyme called topoisomerase separates the double helix above the replication fork and removes the twist formed during the unwinding process. Each of the separated DNA strands function as a template. Formation of RNA primer. An RNA primer is a short segment of RNA nucleotides. The primer is synthesized by the DNA template close to the origin of the replication site. Synthesis of new complementary strand from the parent strand. After the formation of the RNA primer, nucleotides are added with the help of an enzyme, DNA polymerase, and a new complementary strand of DNA is formed from each of the parent strand. The synthesis is unidirectional. In one strand, the daughter strand is synthesized as a continuous strand, which is called leading strand. In the other strand, short segments of DNA are synthesized. This strand is called lagging strand. The short segments of DNA are called Okazi fragments. The fragments are joined together by the enzyme DNA ligase. The replication stops when the replication fork of the two sites meet at a site called terminus, which is situated opposite to origin of replication site.